In this video, we will discuss about therapeutic efficacy of CRISPR engineered human CAR T cells. In the previous video, we have discussed the results of this CRISPR engineered CAR T cells in the mouse model, which is also a kind of in vivo system. But here they wants to check the system for humanized CAR T cell model. So for that, what they did is they first make the humanized CAR T cells with NRA2 that can promote interleukin 12 and did the in vitro studies or stimulate these cells in the in vitro system, and they find out that in vitro system are able to form the cytokines, not only this IFN-gamma interleukin 12 but also this TNF cytokines. So once getting a positive outcome from the in vitro studies, they also did the in vivo human CAR T cell models with interleukin 12. So for the first case, they use the same system, that is NRA2 with interleukin 12, and they are able to see tumor suppressions and also increased survival rate. And the different cytokines results are also shown. For example, IFN-gamma is more in the case where interleukin 12 is there with NRA2, and the granzyme B is also high, and this humanized CAR T cells model also shows a high proliferation markers. And thereafter they check or compare the gene expressions between the tumor and the spleen. And one more thing that they are able to find out is that the CD8 plus and CD4 plus T cells are high in both the spleen, blood, and also in the tumors. So now they wanted to check the intratumoral CAR T cells gene expressions in the spleen and also in the tumors, and they are able to see a difference in the gene expressions, and and most of these upregulated gene expressions in the tumors are related to this interleukin 12, TNF signalings, this STAT4 signalings, and based on this upregulated genes, and then they use the gene set enrichment analysis to find out what are the pathways that are upregulated in the. So when they compare it with the NRA2 with IL-12 and NRA2 knockdown, they are able to see a high TNF A signalings, G2M checkpoint signalings, E2F target signalings. And however, when they compare it the interleukin 12 with MOC, the similar kind of significant upregulations they are able to see. But when they compare it with the NRA2 knockout and MOC, they do not see significant upregulations that can be seen here. So so once validating that their system in human engineered CAR T cells with IL-2 and works well and has a better tumor suppressions or therapeutic efficacy, they want to go one step forward and check the system with interleukin 12, as some of the cancer models requires interleukin 2 expression. So first they check it with interleukin 12, now they check it with interleukin 2. In that case, they see a modest improvement in the tumor suppressions compared to the case where interleukin 2 is not there, and also they see a modest PD1 interleukin 2 improvement. So in that case, now they wanted to in order to improve this tumor suppressions and PD1 interleukin 2 interactions, what they did is they changed the promoter to the another candidate that they have identified in the figure 2, or results that are presented in the figure 2, that is RGS16. When they use the RGS16 with interleukin 2, they are able to see a significant suppressions in the in the tumors. At the same time, high expressions of the proliferation markers in the RGS16 interleukin 2, and also the high expressions of the CAR T cells in the tumors and spleen compared to its control, that is MOC and RGS16 knockout case. So this results further indicates that their human CAR T cells that are engineered with CRISPR knock-in with different cytokines can be used for therapeutic purpose or the clinical for the therapeutic purpose, and it has a better therapeutic efficiencies with different cytokines, as they have shown the results for interleukin 12 and interleukin 2. So I hope you understood how they showed their therapeutic efficacies of the CRISPR engineered human CAR T cells. We will discuss further results related to the same topic in our next video. So because this is just a half part of the main figure, we will discuss the next half part in our next videos. That's all for this video. If you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. Thank you for your time.